Welcome to this explainer, where we're going to dive into a massive biological mystery, one that completely challenges our basic understanding of how the human body actually communicates. I mean, have you ever stopped to wonder how a tiny, almost impossibly small dose of a medicine can somehow affect your entire body? If you've ever just marveled at how the tiniest of signals can orchestrate these massive changes in our biology, well, you are definitely in the right place.
So we call this the Picogram paradox. It's essentially the story of how microscopic signals, specifically these microdose plasma proteins, somehow manage to trigger huge physiological changes across vast circulatory distances. And the craziest part? They do it without getting completely lost in the noise.
To navigate all this complex biophysics, here's our roadmap. We'll start with the paradox itself, look at why the math says it shouldn't work, and then break down nature's incredible three-stage solution: physical forces, chemical bridging, and biological amplification. Finally, we'll wrap up with what this means for the future of therapeutics.
Okay, Section 1, the Picogram paradox. Let's start with a biological rule we've pretty much all accepted and see exactly where it breaks down.
Standard biology textbooks will tell you that organs secrete proteins into the vast bloodstream to trigger target cells. Think of something like the liver sending a signal all the way to the bone marrow. Sounds simple, right? But think about the sheer scale of our circulatory system. At picogram concentrations, which are incredibly tiny, we're talking less than 100 picograms per milliliter, the vastness of our blood network makes a random collision between a signal and a receptor statistically improbable. Nature just isn't throwing darts in the dark here.
Moving to Section 2, the mathematical impossibility. Because what's really fascinating here is how the actual math completely shatters our whole random mixing paradigm. Let's run the numbers.
When you factor in Avogadro's constant, molecular weight sitting around 20 kilodaltons, and the fluid volumes in a standard 10 micrometer capillary segment, the math reveals something genuinely shocking. In this rushing river of blood, there are barely any target molecules present in a given microsegment.
Look at that number. You only have 0.03 molecules per cubic micrometer. That is effectively zero. To put that into perspective, in a standard 100 micrometer stretch of a capillary, you might have roughly 3 target molecules floating by. Just 3. That makes random collisions practically impossible. It proves, beyond a doubt, that physics demands a non-random targeting mechanism. And that impossibility perfectly sets up the solution. Nature abandons random chance entirely for a highly engineered, 3-stage biophysical funnel. We're talking about a multi-scale sequence here, from physical concentration to chemical stabilization, all the way to intracellular ignition.
So let's see how this builds, step by step. We start with physical forces that sort the blood, move to chemical locks that secure the signal, and end up with a massive biological explosion inside the cell.
All right, Section 3, Stage 1, Physical Forces. Our very first step to solving this impossibility relies purely on the sheer physical dynamics happening inside your blood vessels right now. It all starts with flow.
Consider the hemodynamics acting as a physical sorter. Because of something called laminar flow, heavier masses are kept right in the fast-moving center current. Meanwhile, the smaller proteins get physically marginalized. They're pushed to the slow-moving edges. This dramatically increases their local concentration, right exactly where they need to be, hugging the walls near the target tissues.
I absolutely love this analogy from the source material. Imagine red blood cells as these big fish holding the fast center lane of a river. Just by being there, they force those rare, tiny micromolecules out into the slower marginal zones near the vessel walls. It's such an elegant, totally passive sorting mechanism.
But it gets better. Once they're at those slow-moving edges, the negative charge of the capillary wall acts like a giant Velcro net. It snags these rare, free-floating, positively charged proteins. This halts their momentum entirely and positions them directly adjacent to the cellular receptors. Boom! The physics have done their job. The molecule has officially arrived.
Which brings us to Section 4, Stage 2, Chemical Bridging. Catching the molecules is great, but it isn't enough. We need chemistry to actually lock them down so the signal gets transmitted.
Now, unlike standard pathways where things might just casually bump into a receptor, these ultra-low concentration pathways are highly engineered. Take the FGF19 and 21 pathway, for example. It relies on a mandatory helper, a co-receptor known as beta-klotho. Its job is to catch, mechanically twist, and physically bridge that rare signal. If you knock out that beta-klotho lock, The signal is abolished entirely, no matter what the dosage is. It acts as a strict guarantee for transduction.
And we actually see this exact same precise mechanical requirement mirrored in completely different systems too. Take the exceptionally low concentration TPO pathway. That's the signal traveling all the way from the liver to bone marrow stem cells. The underlying mechanics are remarkably consistent, even across totally divergent physiological systems. Whether we're looking at metabolic pathways using FGF complexes or blood cell regeneration using TPO and MPL receptors, both systems pair an impossibly low signal volume with highly restrictive, localized receptor complexes to stabilize the message. It's really a universal blueprint for distal signaling. And this chemical dependency? It is absolute. Researchers noted that if you sever the distal, liver-derived TPO source, local cells are completely unable to trigger activation. While your endogenous local TPO keeps a baseline cellularity, without that specific distal signal, downstream blood cell production completely collapses. It just goes to show that this microscopic distal signal is a hard physiological requirement. It's definitely not just a backup system. On to Section 5, Stage 3, Biological Amplification. Okay, we've caught the molecule, we've locked it in place. Now, we just need the spark. That's the final stage.
It's literally like turning a key in a car ignition. This single, stabilized molecular event unleashes an intracellular cascade that amplifies the initial signal by more than 10,000 times at each tier. The tiny picogram protein isn't actually providing the raw energy itself. Rather, it's merely releasing the engineered constraints of the local biological network. It provides the biological horsepower needed for a massive whole-cell response.
Finally, Section 6, the blueprint for therapeutics. So, after all that, what's the big takeaway from this intricate journey across all these different scales? And more importantly, how do we actually use this?
Well, the picogram paradox is officially resolved. Distal signaling is absolutely not a random chemical mix. Instead, it's a brilliantly engineered sequence. Fluid dynamics and electrostatics guide the way, structural chemistry locks the signal in, and biological amplification scales it all up. It is a complete, masterfully designed biophysical blueprint.
Which leaves us with this. If Mother Nature utilizes this exact multi-scale blueprint to target stem cells with such microscopic precision, the ultimate question is, how can we harness this architecture? Think about it. We could build the next generation of life-saving therapeutics. Imagine engineering drugs that flow, snag, and ignite exactly like this, targeting only the cells that need them without flooding the rest of the body. The possibilities here are genuinely boundless. Thanks so much for joining me on this explainer, and hey, keep questioning the impossibilities.